Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you not one, but two really fascinating guests today involved in creating a better tomorrow on some really unique fronts. We are joined today first by Michael Poisel, who is the Executive Director of PCI Ventures, which is a division of the Penn Center for Innovation here in Philadelphia, which is specifically focused on creating early stage businesses founded on University of Pennsylvania technologies. He is responsible for managing PCI Ventures, including transitioning technologies into startup companies, assisting with the principal investigators with funding strategies, including grant applications, ultimately as well as mentoring these developing companies. Prior to UPenn, Michael was involved in investments in enterprise software, business services for NewSpring Capital, Apax Partners, and G Capital. We had a 10-year career in private equity. He began his career in manufacturing, working at General Electric and Lockheed Martin. He graduated with honors in mechanical engineering from Rose-Hulman Institute of Technology, holds a master's in systems engineering from the Moore School of Engineering of UPenn, also has an M.B.A. in finance and entrepreneurial management from Wharton. And additionally, he serves as the chief executive officer of the PCI Ventures spinout PhylloPharma, which we'll be discussing today, which is a really unique company developing novel plant-based technology platforms for cost-effective and shelf-stable protein delivery. We also have the honor today of being joined by Dr. Andreea-Madalina Serban, who serves as PhylloPharma's Chief Operating Officer. Dr. Cerban is a medical doctor by training, specialized in pediatric surgery, a medical degree from Davila University of Medicine and Pharmacy in Bucharest. She also has a master's degree in business administration, focused on behavioral economics from University of Bucharest, as well as a master's in genetics from University of Paris, and a master's in regeneration, replacement, and tissue repair, surgical sciences from Paris University. Really unique background, some interesting topics we're going to be getting into today. But Andrea, Michael, thank you so much for taking the time to come on the show today to talk a little bit about these teams.
Thank you, Ira.
Thank you so much for inviting us, Ira.
Yeah, it's great having both of you. I would love to start off, as we typically do, by handing the floor to both of you independently. So we can start with Michael and then move to Andrea. Mike, take us just a little bit more into your story and a little bit about this current role in terms of, I know we refer to the Philadelphia ecosystem now sort of as this Silicon Valley when it comes to all things biomedical, but talk a little bit about your background and what got you interested in sort of moving from, as I mentioned, sort of the enterprise software business to sort of managing this really interesting, diverse portfolio of technologies coming out of UPenn.
Certainly. Thank you, Ira. So I spent 10 years in venture capital, most recently with NewSpring Capital here in Philadelphia. And we had a very successful run. The firm was doing great. But as with any firm, as they get more successful, you raise larger funds, you start to transition the strategy of the firm, and it becomes later and later stage. And when the opportunity at Penn came along, I was very interested in being able to help the university with early stage technology and trying to transition that technology into a commercial setting. And that's really where this all started. When I joined Penn, I saw too many examples where technology would get to the point of publishing a paper, but there wasn't a clear path as to where it would go from there to actually being able to try to make it a commercial product. We certainly had a licensing strategy, as did most other major universities, but for many reasons, that strategy was being challenged, certainly in part because large corporations weren't employing researchers as much as they were previously. They were going more to a model of just D rather than R&D, which then made it harder for the university to be able to license patents that were basic in nature. So we really needed a strategy for advancing these technologies to a point that they would look more like products that companies would want to develop further. And that's where this was all born from. And I came to university to help develop that process, which is what we have today. So within PCI Ventures, we have different entrepreneurial programs. The one that's most notable is called Upstart, where we provide a high level of services to the companies to be able to help them get to a point of success.
Excellent. And Andrea, I read through your bio, but I dove a little into your scientific publication history, and you have a fascinating array of things that you've been focusing on over the years. I mentioned sort of the pediatric stuff, and you've published on mitochondrial diseases and congenital abnormalities, but at the same time, you have papers on sort of phytopharmaceutical compound discovery. You have work in stem cell transdifferentiation, in pancreas regeneration. Talk a little bit about yourself, because you have really a very unique mix, and I can see why. Ultimately, we'll talk about PhylloPharma, why you're perfect for what you're doing there, but take us into your past, please.
Thank you so much, Ira, and thank you for taking the time to actually review all my portfolio. I would consider myself to be very lucky. I had a lot of opportunities with time. So it all started with me going to the medical school in Bucharest, as you've mentioned. I decided to continue training in pediatric surgery. I wanted a specialty that would lead to results and lead to visible results fast. So that was, to me, the specialty that could bring, as a clinician, the highest impact. I did my training in Bucharest. Then I went on to do some fellowships in different places in Europe. I was fortunate to work at Great Ormond Street Hospital in London. I also worked at Necker Enfant Malade Hospital in Paris, and I joined the team at Canal Clinic in Luxembourg. So those were all excellent experiences that allowed me to understand the ups and downs of a clinical career from a perspective of a person who wants to see impact. Besides my clinical training, I've also always been passionate about research. Again, because I wanted to be a factor of change, and I felt that I could be one. So in terms of research, I had, as you've mentioned previously, I've done a master in genetics at the René Descartes University in Paris. That was back in 2012, so quite a while ago. Then 2 years later, I was extremely fortunate to receive the French government scholarship for a Master of Research at Pasteur Institute in Paris. And this is when I was initially exposed to regenerative medicine, cell therapy, so an extremely fascinating topic of research. Then I came back to Romania for a few years, and I joined a team at the Center for Excellence in Translational Medicine. And this is where I was actually where I've worked on the research project that was closest to my heart. It was a research project funded by the European Union, and it was a collaboration with Cerban Medical Institute in Israel. And this was a project on replacement therapies in diabetes, which also right now with our project at PhylloPharma fits very well into my research interest. And on the entrepreneurial side, again, going back to impact and bringing transformation, I got my M.B.A. in 2019, and I went down to founding 2 companies in Europe. In 2021, I was selected to represent Romania for an entrepreneurial program that was supported by the US State Department. And that's how I joined Michael's team at PCIV. And this is where I learned more about Dr. Daniel's technology developed at University of Pennsylvania. And that's how it all started. So I joined the team as Chief Operating Officer, and it's been a great adventure ever since.
Oh, yeah. Oh yeah, no, most definitely, and I very much enjoy going through PubMed and looking at sort of that journey as it was unfolding. Yeah, I mean, let's start off, and we can go both, if, Mike, from the perspective of sort of, looking at molecular farming as a business, but then also sort of the history of molecular farming here. And Andrew might want to start with this one, but and just as a sort of disclosure, I've had some of my own business experience a couple decades ago in this space when it was kind of embryonic. But when we look at this area of molecular farming, or sort of the production of non-traditional items in sort of plant bioreactors, whether it's a tomato plant or tobacco, what have you. About 20 years ago or so, the space was sort of heavily focused on, how we could use this interesting biomass to make, at the time, was all these monoclonal antibodies that were in development. So we were afraid that we didn't have capacity, and we needed other systems besides the CHO cells and everything. And about 99% of the industry was focused there. About 1% was focused on sort of the edible vaccines and all the other stuff. It seems like now in 2023, things are the other way, that, maybe plants didn't pan out as great bioreactors to make those drugs, but they're good for a lot of other things. Talk a little bit, if you would. Maybe let's start here with Andrea, and you mentioned Dr. Danielle, because that's where a lot of this began at Penn. Talk a little bit about just the history of and the evolution of molecular farming per the Daniel Lab and University of Pennsylvania in general.
Sure, thank you, Ira. So it's fascinating. What Dr. Daniel has managed to do at UPenn is life-changing, and I think it will be the next breakthrough in pharmacology. So he uses genetics. He basically isolates the gene of interest and inserts that gene into the chloroplast. This, as simple as it sounds, it can change the way we see drugs currently. So he has managed to do this for proteins of different sizes, starting with 7 residues up to several thousand residues, like 3,000, 4,000. So as mentioned before, he isolates the gene of interest. He inserts that gene into the plant leaves. He uses lettuce for that, because lettuce has the advantage of allowing rapid water removal through lyophilization. The powder that you obtain through that process is shelf-stable, so you can keep that at room temperature for up to 2 or maybe even more years. So we can see the huge benefits there. So he uses a gene gun. He inserts the gene of interest into the chloroplast. He grows the plants. The plants give modified seeds. The modified seeds can be later used to obtain other modified plants. So the process is very easy, seems very easy at least. Once you get the mature plant, you put that plant through a lyophilization process, you obtain a powder, and then the powder can be used for either topical treatment or systemic effects. When we talk about topical treatment, we would put that powder into a medicated chewing gum, and the medicated chewing gum can decrease the viral load for different viruses that have a reservoir in the oral cavity. We know for sure SARS-CoV-2 has his sweet spot of replication in the salivary glands. We know it's true for influenza viruses, HPV, different herpes viruses. So there is a whole range of viruses that have an oral reservoir. So when we talk about topical treatment, we talked about the medicated chewing gum that will go in the oral cavity, in the epithelium, and either block the virus from entering the cells, or it will even block the viruses before having any contact with the oral mucosa. And we said there's a second part to what Dr. Daniel has managed to do, which is going for systemic effects, which means that you would take the powder that you obtain through the lyophilization process, you will take that powder containing the protein of interest, you will put that into a capsule. The capsules, once ingested, will be protected by the acid in the stomach. We know that this has always been the problem with proteins. We can't orally deliver proteins because they would be destroyed by the enzymes in the gastric acid. So you would put that into a capsule. The plant cell walls protect the protein. The protein reaches the intestinal portion that follows the duodenum, so just below the duodenum. The bacteria that's localized there would break the beta linkages in the plant cell walls, will release the protein of interest. Dr. Daniels uses CTB. So the CTB will bind to those specific receptors present in that area. They will be later cleaved, and the protein of interest will be released in the bloodstream. And this systemic administration, for us, the holy grail is insulin. And this is one of the projects that we are developing, we're working on.
And just while we're at it, can you say a few other words about sort of the unique, because you mentioned the chloroplast expression, and then obviously the major issue with therapeutic proteins and the destruction. Talk about what we've learned again over the recent years in terms of the benefits of, let's say, the plant biology that make that biomass source such a so useful, let's say, as not just a production system, but a delivery vehicle as well.
Yeah, we talked about delivery, and we talked about the fact that the plants allow for the protein to be protected. So that's the huge benefit compared to any other delivery systems. Finally, we have a way of making a protein that is orally ingested, reach its target in the digestive tract, where the receptors are, and allow that protein to finally reach the bloodstream.
Excellent. And as you mentioned, the chewing gum project, and that's been in the news a lot lately. So here specifically, you were looking to, I believe, take ACE receptor protein, express it, and then ultimately put it in the form per the plant and then into the chewing gum vehicle, so that if there is virus, and this virus loves, unfortunately, the ACE2 receptor in our bodies, but if we can bind it ahead of time in the oral cavity. Take us just a little bit of what's going on with this project. I know there's a clinical trials plan to ultimately study this, I think in combination with the School of Dental Medicine at Penn, but talk a little bit about sort of the history of this particular project per COVID, and where it's gone so far.
Sure. So we were lucky that Dr. Daniel has worked for decades prior to COVID existing on ACE2. He had investigated the effects of ACE2. He had the technology ready to isolate the gene of interest and insert it into plants. So once the entire world knew that COVID loves ACE2, as you pointed out, yeah, the obvious direction to move his research was towards exploring ways of fighting the virus. So the way he does this is in two mechanisms. Once the virus would be trapped because the ACE2 would bind to the spike protein, as you mentioned, that's located on the surface of the SARS-CoV-2 virus. And as a second mechanism to block the entry of the virus in the oral mucosa, you have the GM1 receptor. And the GM1 receptor will block the virus cannot enter the oral mucosa because it will be blocked at the level of the epithelium. In terms of development stages so far, so Dr. Daniel back in May had managed to obtain FDA approval for a phase 1/2 clinical trial. We know ACE2 from before. We have ACE2. We all have ACE2 in our bodies. So we know that this is a safe component to administer. So that allows the clinical study to go beyond what we will typically see as a phase 1 of exploring safety. We have a phase 1/2 clinical trial that has been approved by the FDA, allowing us to first, of course, explore any adverse effects of the substance, but also move beyond that and be able to prove a reduction of the viral load. So we would take saliva samples from the patients before and after chewing the gum, and we would determine the viral load there. So the FDA approved this again back in May, and there have been several steps that needed to be followed in order to have the clinical study started, but hopefully it will soon enroll patients. So
excellent.
We look forward to that.
Excellent. Mike, moving over to you, and we'll come back to Andrea to discuss insulin and some other projects. Clearly, a project like PhylloPharma, and it's many things, right? It's a biotech company, it's a biopharma company, it's a device company, a biomanufacturing. Talk from a PCI perspective. You look at this, you have amazing technology coming out of the laboratories at Penn. Talk a little bit how you go about what you do in terms of, hey, we have this tool. Let's set up a company. Let's get it funded. Let's create this initial business plan and clinical plan, what have you. Talk about your role here and a little bit of the history on how you put together sort of what exists today as sort of the corporate model per PhylloPharma at PCI.
Sure. When we originally began the program, we saw a need for a mechanism that faculty could take their technology and apply for SBIR grants, which are small business innovative research grants funded by the government. And so the original idea was, could we form a paper company for the purpose of applying for that SBIR, which we did, and we started to have some success. Through that process, I met the director of the National Science Foundation, director of SBIRs, and became friends with him. And he really was interested by the model, and he wanted to get the perspective of all of the SBIR program managers. So he arranged an opportunity at a conference where I got to sit down with all of them and described what I was doing. And I was very much in fear that they would say, well, you're really screwing up the program because you're just forming paper companies, right?
Yeah.
And they said, no, we do appreciate that because we don't have enough good proposals. So we like that you've created a mechanism for Penn professors to be able to do this. But they pushed me and said, could you do this on a bigger scale? Could you actually create a real company, not just form the paper and sort of stop there?
Right.
And I took that back and started to think about how could we develop a process to generate an actual real company. And for us at Penn, because we do not have a fund, we don't put initial funding into these businesses, we really need to bring on a CEO early in the process and use them and work with them to raise the money and develop the plan for the business. And I say plan for the business instead of business plan because we don't write 30, 50, 100-page documents anymore, but we need to know where we're going and what's the vision for the business. And so we bring the entrepreneur on board, we give them equity in the company, and we work with them to develop what the company will be. So I give the example, many times I will ask a faculty member to tell me about their company, and they'll start talking about their technology. And I'll say, okay, understand that's the technology. Now we need to figure out what is the business we're going to create from the technology. And really work with the management team to understand what is the market opportunity. First of all, what's the problem we're trying to solve, right? What's the market opportunity and what's our solution and how is it going to be defensible? And really those kind of basic ideas about starting a company and going from there. And we work with them every step of the way. So bringing on the management team, helping them come up with the plan for the business, talk about the funding strategy. We have a Rolodex of investors that we can bring the company to and talk to them about the opportunity in front of them. And then once it looks like the company is going to get funded, 6 years ago, we opened the Pennovation Center, which is an incubator where companies can come and build their businesses. We filled that building. We asked Penn to renovate a second building. They did. We filled that. Our third building came online last January. It's now full. And so Penn has commissioned the fourth and fifth buildings to be built, and they'll break ground next spring.
Excellent.
And then from there, just to finish that thought, somebody from my team stays on the board of the company as long as they want us to be. And that's an unusual feature of this program, that we're that intimate. I describe it as we're not cheerleading from the sideline. We're actually in the game. We're working with these companies on almost a daily basis, helping them solve their problems and reduce the friction of starting a business. That's really where I see one of our core values is just helping companies reduce the friction that they inevitably will experience through this, and work with them as closely as they can. If investors no longer want us to be a part of things, they want us to back away, we'll certainly do that. We have lots of flow that keeps coming in. A new pipeline of companies is always waiting for us. But sadly for us and our time, we've only had that happen a couple times. And so we are on the boards of a lot of companies and working with a lot of different ones at the same time, which is fun because we get to experience so many different types of technology.
Absolutely. Andrea, we mentioned your own work in diabetes and the pancreas and stem cell transdifferentiation and things like that. Clearly, one of the focuses sort of after the antiviral chewing gum program is on insulin, and obviously the oral insulin has always been one of those holy grails of the industry in general. Take us a little sort of where you're going, or where you hope to be going with either that or related portfolio items in sort of the metabolic space.
Sure. As we mentioned initially, there are two major types of applications for Dr. Daniel technology, the topical treatment and the systemic one. So we spoke about topical treatment as it relates to the ACE2 medicated chewing gum. With regards to the systemic effects, oral insulin, or to be more specific, oral proinsulin is one of the projects we will focus in the next few months. What does that mean? We know at the moment the only option for administration of insulin is through injections. This comes with high costs. This comes with the needs of storage at storage in specific conditions that make the insulin unavailable for a large portion of the population out there. And it comes with discomfort for the patient. We know it often happens that there are complications at the site of the injections. It's obviously not easy for all categories of patients to administer injections. So we try to help patients with diabetes, but right now there are a lot of limitations to what we can do with injectable insulin. Sure. To add to that, we know the risk of hypoglycemia. That's so significant with patients. We know hypoglycemia is more dangerous to them than hyperglycemia, and it can even lead to convulsions or complications on the neurological side. So what Dr. Daniel manages to do with creating an oral proinsulin is, first of all, mimicking as close as possible what we would have in a physiological scenario. So we know in our bodies, if our pancreas is healthy, we would get at a certain stage in the process of obtaining insulin, we would get proinsulin that contains both C-peptide and insulin. And this has been proven to be beneficial because C-peptide has a role in protecting the kidneys of patients with diabetes, the brain, retina. So we know that now. We did not know that 50 years ago when injectable insulin was developed. So with his project, Dr. Daniel manages to obtain an insulin that's closer to a physiologically secreted insulin. And this helps because we expect to have a profile, a pharmacodynamic and pharmacokinetic profile that's closer to physiologic, preventing some of the complications that we have with the current injectable insulin. And obviously, on top of that, we get all the benefits that the platform has to offer in terms of shelf stability for components at room temperature, and obviously oral administration instead of injectable. So we're very excited for that project, as you can probably see.
Yeah, absolutely. And it got me thinking also about, in terms of this unique expression system, you're not limited to one thing. I mean, obviously you could, multiplex and all that. Any interest? I mean, obviously it's early on in the process, but when you think of especially some of these chronic diseases where we learn the sort of the polypharmaceutical nature of things, any interesting sort of brainstorming about sort of combination opportunities, whether it's in diabetes or any of these different conditions where, hey, we now have we have something here where it doesn't just have to be a single magic bullet, a la how our pharma industry has traditionally thought the last 100 years. But hey, we can experiment now with multiple modalities in one chloroplast or in one piece of lettuce and explore some of these polypharmaceutical, because I'm thinking back, and I apologize again for thinking about some of your publications when you were working with sort of the phytopharmaceutical side of things. Those are really complex biochemical cocktails that you're supplying in dementia and some of these other studies you've done. Any interesting things on that front that you could talk about?
Well, obviously for us, it's very exciting to look at this type of tool because, as you've mentioned, it in theory provides unlimited options to develop any sort of proteins. We've mentioned that Dr. Daniel has done it already with different components from very small ones to bigger ones. So he has developed plans for quite a few proteins already. But at the moment, we as a company, we decide to focus on the ACE2 product,
Sure.
on frill and insulin. So we will definitely explore what this platform can offer in the future. And you're right. In theory, we could explore as complex as possible, but we'll see where
Yeah.
this goes.
Got it. Mike, what's and you can tag team on this one, but what's coming up that's hot for the company, either financially or otherwise, for 2023, funding rounds, conferences, upcoming publications you can mention, please.
Yes.
You can tag team on that one as you wish.
We have several things going on, all in motion at the same time. It's an exciting time for the company. Obviously, we are in the process of talking to investors about our next round of funding. Dr. Daniel will have a paper that will be published soon that we expect will, of course, garner quite a bit of PR about. There's some significant findings there, which will be exciting to see, and several other projects going on. We hope to be able to start several of our clinical studies this year. So this will be a significant year in the company's development, to be sure.
Andrea, anything out on the conference scene for you coming up, or places that we're going to see you present? We can meet you face to face. Other things happening, please.
So what excites me most is the research part, and certainly Dr. Daniel's paper is something we all look forward to have it published and to have it out there in the world, because we are sure it will bring a lot of relevant feedback from the scientific community, but also from potential commercial partners.
One last thing while I have you both, and I just, because I know sort of early on in Dr. Daniel's research, he did a lot of work on the antigens and sort of the edible vaccine front. Are you getting a lot of sort of attention in this interesting vaccine world in 2023 for sort of the edible vaccine concept, whether it's here in other countries? I mean, I know that was years ago. I actually met around the corner with Hillary Kaprowski when he was working on rabies vaccine, and I forget, tomatoes or something, but anything interesting that I know it's not probably core at this point to the company, but are you seeing a revitalization of that whole edible vaccine concept at any point in 2023?
Yes.
We've seen other companies pursuing it. I mean, it's not something that we've been focused on right now, but I think there is significant interest in that area. I think the interest in plants as delivery platforms is under significant interest around the world, and we're seeing many other companies doing things that are not the same as this, but certainly working with plants as a platform, get significant funding, and so we certainly see that we're on the cutting edge of a brand new technological trend. And I'll let Andrea add as well.
I fully support what Michael said, and indeed, Dr. Daniel has already expertise in the field of oral vaccines, so who knows, it might be something we will explore as a company.
Excellent.
At some point.
And then just one other thing I'm thinking of as we're talking about this, and obviously we get to scale up and thinking about sort of large scale production here, but we're all here in the Philadelphia area, and right across the river here is our garden state. But clearly, these tools, especially sort of the hydroponic and alternative growing technologies, are getting kind of hot in New Jersey. Are you getting a lot of excitement for sort of the grower community for what's coming down the pike in terms of growing pharmaceutical crops versus just growing crops for calories?
It's definitely a huge and growing industry. There's a lot of different things being grown. During our research, we learned of a company in North Jersey that's growing strawberries hydroponically and selling them for an exorbitant amount of money. Andrea, I don't remember. Do you remember what the price was?
I think $200.
Oh,
yeah. It was incredible amounts of money that people were spending. So you have that extreme of things. You have lettuce being grown hydroponically for high-end stores that want to be able to produce that. And with the outbreaks of listeria and that in some of the farm-grown crops, growing hydroponically has that attraction of avoiding some of the other bacteria or infestations that might be able to happen. So certainly that's an interest as well. And of course, with the legalization of cannabis across the country, you get a lot of the growing operations that are growing cannabis as well. So there are significant infrastructure being built out for growing. It remains to be seen how we'll need of use of it. Obviously, right now, we're leveraging Dr. Daniel, and he has a greenhouse here at the Pennovation campus. And so we want to do everything we can through that mechanism for now. We'll see what the future holds in that regard.
Awesome. Yeah, I like to see, these ecosystems coming together, and it's exciting that, it's happening here in the Philadelphia, Silicon Valley region. So it's just I'm looking forward to watching the progression of this happening in the area, and really just wishing you both much success with it, because it is such an exciting area, and I'm near and dear to my heart personally. But again, for everybody that's going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel, again, you've been listening to Michael Poisel, Chief Executive Officer, and Dr. Andreea-Madalina Serban, Chief Operating Officer of PhylloPharma, doing really amazing things in the development of novel technologies for cost-effective, safe-stable protein drug development, all happening here in the Philadelphia area. Michael, Andrea, I want to thank you both again for taking the time to come on the show and talk to us for a little while about these themes. Obviously, thank you for working on them. And as we like to say here on our show, thanks for helping to create a better tomorrow via the tools and technologies you're developing. Really very fascinating story.
Thank you. I really appreciate the opportunity to talk about it.
Thank you so much, Ara, for allowing us to share our story. It's great meeting you.
Nice meeting both of you.